Hi, everybody. We are going to talk about part two of bacteria. So where we left off, we were just about to talk about prokaryotic nutrition. So we see this great chart here has divided up nutrition by energy source and by carbon source. So the energy source, prokaryotes can either get their energy from the sun or from chemicals, right? They can either absorb their energy from sunlight, or they can absorb it from the potential energy found in various chemicals. And then we have the carbon source. The carbon source is going to be, where does the carbon come from to make all those carbon compounds? It's either CO2 or organic compounds. So this would be like chemicals that are specifically carbon-based chemicals, right? So if we look at this, if we look at the photoautotrophs, these would be bacteria that are very similar to plants. They absorb energy from the sun, and they get their carbon from CO2. But there's also another type of autotroph called a chemoautotroph. A chemoautotroph gets carbon from CO2, but then it can get its energy from the potential energy found in certain chemicals. So these are the two types of autotrophs. Now we have the heterotrophic bacteria. These are going to be getting their energy or their organic compounds from taking in something else, right? So here, the organic compounds are going to give these things carbon, just like how we eat organic compounds for carbon. So photoheterotrophs get energy from the sun and then eat organic compounds. And chemoheterotrophs are going to be getting their energy from chemicals and eating organic compounds. So presumably that will come from the same source in many cases, kind of like how animals eat. So these are the ways that prokaryotes can get their nutrition and energy. They have a pretty big diversity. So they either undergo photosynthesis or consuming organisms, or they can consume other chemicals or get the energy from their chemicals. So there's a big diversity in terms of what prokaryotes can do for nutrition. Another important thing that prokaryotes can do is they can form what's called biofilms. So a biofilm is when a lot of bacteria get together and they undergo what's called quorum sensing. If you remember from semester one, that's when there's enough bacteria together that they form a sticky film that allows them to share resources and sometimes communicate. Plaque on your teeth is a biofilm that is caused by prokaryotes that live there. And when there's enough prokaryotes, they start to form plaque, and the plaque allows them to share chemicals and nutrients, and then also to communicate with one another. Some biofilms can form inside water lines and sewers. So biofilms are pretty common. Bacteria can be helpful. So some bacteria can help us. We use bacteria for sewage treatment to break down a lot of the waste that's produced and put in our sewage. Bacteria can fix nitrogen into the soil, right? So gaseous nitrogen is pretty inert, but bacteria can take it in the soil and turn it into something that plants can use, and that in turn helps us, right? Since we need plants to eat. Bacteria can help us decompose certain things. So we need bacteria as decomposers. That's another important role of bacteria. Here we have an example of nitrogen fixing. So bacteria taking nitrogen from the air and putting that nitrogen into the soil for plants to use so that we and other animals then can eat those plants. Prokaryotes, as we already know, play a vital role in genetic engineering, right? Remember how we use bacteria to make insulin. So because bacteria are so plentiful, because they reproduce so quickly, because they're so simple, and they will uptake DNA, it is easy to use them for genetic engineering. Bacteria can also be what are called pathogens. A pathogen is a disease-causing agent. We often think about bacteria as pathogens, and many bacteria are pathogens. One common pathogen is a staph infection. Staphylococcus is a bacteria that can cause very serious infections, although it's pretty common. If it gets very deep in our tissue, it can cause some serious problems. Staph infections have become resistant to many of our antibiotics over the past few years, which makes them even more dangerous because we don't have a lot of medicines left that can kill those bacterial infections. Others, so anthrax is a bacteria that was in the news about 20 years ago because it was being sent as like in terrorism in spores. What else do we have here? Lyme disease spread by deer ticks is a very serious bacterial infection. The plague caused by bacteria living in rats. Salmonella found in unsanitary food conditions. These are all types of pathogenic bacteria. Now, one last thing that we need to learn about bacteria is the idea of horizontal gene transfer. And this is going to be really important because this is what keeps genetic diversity in something that undergoes asexual reproduction, right? For animals and many plants, genetic diversity goes hand in hand with reproduction, right? We mix up the genes in the next generation. So we have vertical gene transfer, but bacteria do not. Bacteria reproduce, and their genetic variation, that's two separate things. So genetic reproduction, mutation, and genetic recombination are all going to promote genetic diversity in prokaryotes, but it's different from prokaryotic reproduction. So here we have bacteria reproducing asexually and producing clones, right? Bacteria can reproduce very quickly because it's asexual. There are mutations that happen. So mutations do occur, and that causes some genetic variation. Prokaryotes are haploid, which means that when there's a mutation, it is immediately subject to natural selection. There's no hidden recessive genes in a prokaryote, and that's because prokaryotes are haploid. So if bacteria reproduce asexually, then how is it that they can change over time? Besides mutation, there are three mechanisms of horizontal gene transfer that we actually already know about. So this should be reviewed. Transformation, that's picking up random pieces of DNA. Transduction, that's infection via a virus. And then conjugation, this is when two bacteria come together and exchange plasmids. So transformation, just like the lab we did, the pGlow lab, bacterial cells pick up random pieces of DNA that they find, and they incorporate it into their plasmids, and then they can express this. This might seem like a bad idea if you think about it, and in some cases it probably is a bad idea, but the genetic variation is worth the risk of expressing these strange pieces of DNA. This is what we take advantage of to put insulin into a bacterial cell and to force a bacterial cell to make insulin. Then there's transduction. This involves a bacteriophage, which is a virus that infects bacteria. Bacteriophages will inject their DNA, and then they will force the bacteria to make more viruses, right? Sometimes, though, when that happens, what the virus will do is it will chop up the host DNA, and sometimes a mistake happens where some of that bacterial DNA that's just been chopped up makes it onto the viral DNA. And so when the virus goes to infect another bacteria, it accidentally lets in a little piece of the bacterial DNA. And if this bacteria survives the virus attack, then this little piece of bacterial DNA can get stuck in here and become part of the bacteria's genome. So it's a little bit sort of almost like crossing over, except with a virus. And then we have conjugation. Conjugation is a really important one. Bacteria have these structures called pili that can form a tunnel between two bacteria. And so what can happen is the bacteria will copy the DNA. The DNA will move through the pili, and it will go into the new bacteria. So if this bacteria on the left has a plasmid that allows it to be resistant to ampicillin, it can be transferred over to the next bacteria. Here we have a diagram showing conjugation, the plasmid copying itself and moving it into the bacterial cell on the right. Right? Another mechanism of horizontal gene transfer, mixing up genetic variations, but not doing it through reproduction. And that's all we have for now. If you have any questions, let me know.